How are you guys? It's been a long time since I've been live. It's actually not that long. I was just live earlier this week. Hopefully you guys are good. For today's session, we're just going to be doing the November past paper. So if you're interested in doing that, they just got released, so that's exciting. If you're interested in doing that, please do stick along. We're going to go ahead and get started when everyone's ready to join. So we give you guys a couple of seconds. But we're going to be doing a bio past paper because the bio crash course are going to continue this weekend. I'm just going to go ahead and grab some chapstick while you guys join so my lips don't get too dry. But how is everyone? Hopefully everyone's good. You guys have been having a good week so far. If you guys are old crash course students, or current crash course students, hi, welcome back. Hi everyone. How are you guys? Are you guys ready to do this November past paper? Hopefully you are. And hopefully if you did the crash course last weekend with us, these SL questions will be easy. So today we're going to be doing an SL bio because of the crash course that we're going to continue tomorrow literally. We're going to continue with day 3 and day 4. So if you're interested in joining, do go check out our crash courses. I'm going to go ahead and get started. So as I said, this is the biology standard level paper 1A. Finally, they've uploaded the November 2025 one. So it'll be really good to do this as practice. And to kind of like compare what they asked in May 2025 versus what they've asked in November 2025. And what I've been doing actually these past few days since they've been released is I've been analyzing the past papers for the crash course students so that they're aware of like the type of questions and so we can make prediction papers and so on for you guys. And if you guys know, and if you've been keeping up with our TikToks, you know that our prediction papers have actually been quite accurate. Okay, so let's go ahead and get started. Hopefully you guys are good and ready to go. The first question just says, which characteristics of the ringed seal is a consequence of the thermal conductivity of water? Will there be a new crash course in April? Good question. No. So the crash course happens once a session. So you have a one session biology crash course, one session chemistry crash course, and one session math crash course. But the thing is that you can, of course, watch the recordings. So the live session only happens once, but you can watch the recordings, um, you know, uh, and you have access to the recordings for the next 2 years. So you can watch the recordings wherever. Most of our students actually, not most, but some of our students actually just buy the crash course just to watch the recordings. So hopefully that will still be helpful for you. All right, are you guys ready to go ahead and start? Some people are saying C in the chat. That's nice. Okay, so C is the correct answer. And we've just covered this exact example in the crash course, and it's so comforting to be like to have it's so comforting to have told the May 2025 students to focus on this example, and it's showing up. So if you guys don't know, for the water units, you don't just need to know the characteristics of water, but you need to know two specific examples. And we've talked about those two examples in the crash course. One of them is the ring seal, and the other one is the black-throated bird. I think that's what it's called off the top of my head. Okay, so those are the two examples that you need to know for water, and you need to know the characteristics of those two animals for water specifically. So if your teacher just taught you about characteristics of water, the hydrogen bonding, the covalent bonding, the polarity of water, but hasn't talked about those examples, you're missing out on something, okay? And so we've talked about this example in the crash course. By the way, if anyone's done the crash course over the weekend and has any advice or like wants to give like their honest, true opinion in the chat for the people who are like kind of iffy about buying the crash course, please go ahead and do like this is not going to be sponsored or anything. So just give your honest opinion about the crash course if you liked it, if it has been helpful, and so on, so that more people can learn about the crash course. If you've chosen C, good job. C is the correct answer. So essentially what you need to know is that water has high thermal conductivity in comparison to air. And the key idea is when the water gets really cold for the seal, that's bad because then the seal can't maintain a homeostasis. That's my drawn seal, by the way. And the seal can't maintain homeostasis, and the seal's body temperature gets really cold. So what ends up happening is that you have this thick layer of fat called blubber, and it's sort of just an insulating layer that surrounds the seal, and they're able to protect themselves from the fact that water has very high thermal conductivity. Okay, so you don't only need to know about the thermal conductivity and those specific properties of water. You also need to know specifically in reference to these examples in case they ask you to give an example in paper 2. So keep that in mind. Is this biology? Yes, it is SL biology. Okay, let's go ahead and have a look at this question. We've also covered this in the crash course. It's a really simple question, I think, that has been showing up multiple times throughout the year. So hopefully you guys get this. If you don't worry, that's okay. You still have time. Don't stress about it. But it says which molecule or which molecular diagram shows a phospholipid molecule. So excited for the crash course to resume tomorrow. Yay. I'm so excited as well. Okay, someone put an answer in the chat. Right. So good job. Exactly. So how do you generally identify fatty acids? So basically, you might see it like this, which we actually saw on the May 2025 paper two. That we saw that the IB drew out the full carbon to carbon chain like this. But generally, what you're going to see in paper one is that you're going to see this skeletal structure, and this is basically like zigzags, and these zigzags represent really long carbon hydrogen chains. So whenever you see these two long these zigzag chains, you know that this involves a fatty acid. So keep that in mind. Whenever you notice it's a fatty acid, you can then just start counting. You see here, this has one, two, three fatty acids. And actually, you're supposed to know what this molecule is. This is glycerol attached to three fatty acids. Does anyone know the name that you need to know for this one? Here you can see one, two fatty acids, and then be very careful, guys. Do you notice something here? You see a phosphorus. Okay, this is the phosphorus that's part of the phosphate head. So that's how I know B is my answer, because I see a phosphate head and two fatty acid chains. Very good, Sophia. Sophia said triglyceride. Exactly. Good job. Thank you guys, by the way, for 1.4K likes. I really appreciate it. Should we set maybe a like goal of 10K? Let's see if we can do that. Okay, as a little just practice to make sure you guys do active recall and make sure that you guys are also learning from this live. Hi, Lean. Hi. Can anyone tell me what this is and what A is? Who knows? And hopefully, if there's some crash course students in the chat, do you guys remember what it is? What this type of bonding is called? Yeah, that's what I'm trying to know. Who's here from the crash course? I want to hear you guys' input. Does anyone know what D is showing and what A is showing? Who knows? The assistant to the main tutor is saying that she's doing the resource packs. Hydrogen bond, no. So very be careful. Um, so condensation, dipeptide. You guys are saying correct words, but this is not really what I'm referencing. But you're kind of correct. So for example, this we see here that we have a main carbon, an amino group, and a carboxyl group attached to another essentially main amino group, carboxyl group. And so it's not a dipeptide. Be very careful. You have one, two, three, four amino acids. And be very careful, guys, because it's actually a question before where they've asked you to count how many amino acids are here and asked you to see exactly how many amino acids there are. So this is there is peptide bonds in between this, but you wouldn't get the mark if you just said dipeptide, because di means two. Okay, very good. A is carbohydrate. Good job, guys. And the top one on the top is a glycosidic bond. You are correct. The bond between two sugars is called glycosidic. How do you remember? You know that glyco is like glycogen, glycosidic. But can anyone tell me more specifically? I'm going to test you guys now, and hopefully, if someone's in the crash course, they are able to say this easily. What type of carbon to carbon bonding is this? So, what carbon is attached to what carbon? And therefore, what type of branching do I see here? Who knows? Okay. I took the IB and I got diploma with 34 points. Yay, congrats. How did you find it? Condensation. Da da da. Okay. Almost, very close. Good job, Naz. Exactly. So it is the 1 to 6 bonding. So here I see actually is it 1 to 6? Let me count. So how do I count it? Let me do it with you. You have your carbon 1, you have your carbon 2, your carbon 3, your carbon 4, your carbon 5. Okay, perfect. It is the 1 to 6 bonding. The 1 to 6 bonding, because remember this is your 6 carbon. So here I have my first carbon. How do I know it's my first carbon? You always count to the right of the oxygen. So you locate your oxygen in your glucose molecule and you start counting to the right of it. So here you have your carbon 1 attached to your carbon 6 of a neighboring molecule. So this is known as 1 to 6 bonding. Was it 1 to 6? Exactly, good job. And then the bonding that's between here is actually 1 to 4. So if you said 1 to 4, we're referencing this one. 1 to 6 allows for branching. So hopefully you guys are okay with this. If anyone wants me to repeat, I'm more than happy to. Thank you guys for almost 4K likes. You guys are the best. Which past paper is this? Good question. This is the November 2025 time zone 1 SL. All right, let's have a look at question three. It says, which molecule is paired with the component of the cell membrane that allows it to pass through the membrane? And I think, just to let you guys know, because I think it's helpful to know, this is a common question to get. So make sure you familiarize with this. This is part of the new syllabus. And be very careful for question one. You were using examples that are specific to the new syllabus. So make sure you focus on that and don't just learn the water topic very briefly without going in depth um, with the examples that you need to know when you're explaining water. We know that the May 2025 questions have always started with water, and we see that the November, they're not changing that pattern, but they're actually being a bit more specific, and they're like kind of twisting the knife and asking you to, rather than explain the characteristics of water, like the physical properties of water, like the polarity and so on, they're going further and using an ecological example. Okay? Is this chemistry or bio? This is biology. What are we thinking for question three, guys? This is which molecule is paired with a component of the cell membrane that allows it to pass through that membrane. So I think this is hmm. This is an old syllabus question. Um, but I would say that this is a hard question for question three. Yes, the November papers are out. Thank you guys so much for almost 5K likes. See, okay, I think I'm getting your chats a bit later. See, okay, now I'm seeing it. See, yes, this is the November 2025 paper. That's what we're doing now. C maybe. Okay, good. Sorry guys, I just saw all your messages now. Perfect. This is C is the correct answer. How do we know that? Well, insulin, we know that of course it's not passed through aquaporin. Does anyone know what passes through aquaporin? And hint, it's in the name. Aquaporin, water, very good, Lean, exactly. So water is going to be what's passing through. Good job, exactly. Water is going to be what's passing through aquaporins. Glycogen through a channel protein. Now, why is estradiol passing through the phospholipid bilayer? How do we know this? Who knows? What's the properties of this? And what allows it to pass just directly through the phospholipid bilayer? So if something passes directly through the phospholipid bilayer, what do we know its properties are? Is it hydrophobic or hydrophilic? We know as well CO2 is just a small molecule and it diffuses. How do we know it diffuses? Well, because you need to know the lung unit, and the lung unit basically teaches you that you inhale oxygen and you exhale CO2, and we know that it just passes through like diffusion. I use revision dojo for my IA and told me I would get a 7, but I got a 2. That's so upsetting. I'm so sorry. Hydrophobic, very good job. Exactly, guys. This is November 2025, time zone 1, SL. Exactly. So we know that this is cannot be true. I would connect this to the lung unit. And the reason why this is correct is because we know estradiol is a hydrophobic molecule and therefore can freely diffuse through the phospholipid bilayer. I'm so sorry that that happened to you with your IA. I think we should just stop using. I mean, like I love AI, which I shouldn't because it's really bad for the environment. But I think there's beneficial ways to use it for. What is insulin? Good question. Insulin is a peptide hormone that you should know for your syllabus. You should know for SL, and it's essentially regulates your blood glucose level. Does that, like, for example, have you heard of people with diabetes? Does that ring a bell? When people with diabetes, when they have too much blood sugar in their blood, they need to take extra insulin in order for them to lower their insulin levels. Okay, let's have a read of this question. This is which examples are anabolic and catabolic. This is also common. They've been asking this question in May 2025, so I'm not surprised that they've um, added this question here. Thank you guys for 6K likes. I really appreciate it. Okay, I'm gonna give you guys a couple seconds to think about this, and while you're thinking about this, what were we talking about? Ah, yeah. Um, yeah, I'm so tired of every single TikTok is revision ad. Yeah. Yeah, I think AI can be used beneficial for your studying. I think it'd be really, really helpful. But I just don't like when resources are made using AI. Like I think where we're heading right now is that people are just coding random resources with AI, and I don't think that's very beneficial. I like when resources are made by like teachers. Okay. B, A, maybe. Okay, so we have different answers in the chat. All right. So B is the correct answer. Let's have a look. So anabolic, what does that mean? Who remembers? Anabolic, catabolic. Anabolic means what? Breaking or forming? Anabolic, very goodly. Anabolic is building. Catabolic is breaking. Kata is breaking. Therefore, I just have to be very careful with my terminology. So, hydrolysis. Who remembers what exactly? Very good job, guys. Forming anabolic, catabolic breaking. Good job. Now, what does hydrolysis mean? What does lysis mean? Lysis means cell breaking, okay? Cell bursting. So, hydrolysis actually means we're breaking the cell. We're breaking these, in this case, we're breaking the monomers. And therefore, it's not a building because we said anabolic is building. Glycogen formation. Yes, formation means I'm building. It means I'm anabolic. Hydrolysis of macromolecules into monomers. Yes, catabolic means I'm hydrolyzing, I'm breaking. Therefore, this could be correct. Let's have a look at the other answer options. C says oxidation of substrates in respiration. If I'm oxidizing in respiration, what am I doing? I'm taking glucose and breaking it into ATP and releasing CO2 and water. So actually, respiration as a whole is a catabolic process. So keep that in mind, okay? Because this also showed up in the May 2025, where they've used that as an example. And I'm assuming that they're going to bring it up again because they just loved it so much. Protein synthesis, again, synthesis is not catabolic. Protein synthesis, correct. Glycogen formation, no, this is not a catabolic process. So definitely answer is B. Good job, guys. Thank you guys for 7.5k likes. All right, let's have a look at question 5. This is, yeah, this also showed up on May 2025. So if you've done our past paper analysis for the crash course, or if you've just done analysis yourself and you've done lots of past papers, which is really good, you'll notice as well this pattern. And these patterns are really, really helpful to recognize when you're studying for your exam. Has everyone started studying for their exams? How's everyone feeling? I know some of you, the EE deadline is coming up soon. How are you guys feeling about that? What's going on? Hopefully life is not too stressful right now. Um, I submitted my EE a long time ago. Yeah, it honestly depends. But the final deadline, I think, is on the 15th. Like in August. Whoa. That's crazy good. Good for you. I like that. I like that you prioritized it and got it out of the way. Okay, what are we thinking for question 5? Who knows? Thank you guys for 8k likes. I really appreciate it. And have you guys started studying? How are we finding it? C maybe? Okay. I like that you guessed, even if you don't know. Good job, Dylan. My school submitted our TOKSA and EE animal with some of my A's. Yay, congrats. And are you planning on studying right after you're finished? Or are you planning on studying like kind of in between? Let me know. What is everyone's study plan? Okay, good job, Dylan. Even though you said maybe you got it correct, so be confident in your answer. It says the graph shows the rate of photosynthesis as light intensity increases at 2 different carbon dioxide concentrations. What are the limiting factors of photosynthesis at region X and Y? So I'm going to tell you now because this is what I'm doing for the crash course students and I want to help you guys out so that you guys are able to study. So essentially what they've started adding or what they've focused on, I think, in the syllabus, and what you'll notice if you do the May 2025 as well, is that they started focusing on limiting factors. Okay. And they're trying to see what limits photosynthesis, what limits respiration. And there's this key idea is that if you don't have enough substrates, or if you don't have enough light intensity, if you don't have enough glucose, if you don't have enough I'm talking about respiration, sorry. If you don't have enough CO2, if you don't have, because you know you need CO2 plus water to make glucose, okay? For photosynthesis, if you don't have enough of those substrates, your photosynthesis is going to decrease. If you don't also have enough light intensity, your photosynthesis is going to decrease, okay? And they've just focused on that. So I'm telling you that so you guys can keep it in mind. So when you're reviewing your notes or you're reviewing the crash course videos, whatever you want to do, you keep that in the back of your mind because I think that they're like fixating on that, okay? And that's the most you can do to try to predict the upcoming questions. So what are the limiting factors at regions X and Y? Again, we've seen a question like this on the May 2025, so they're being very repetitive. So we see at X, we have the 0.40% CO2 as the straight line and 0.04% of CO2 at the dotted line. And we see at X, there's different things that are limiting. Okay, because as you start increasing the light intensity, so here on my X axis, as I start increasing the light intensity, what ends up happening, my rate of photosynthesis increases. So here, when I'm having low light intensity, it's basically saying that this low light intensity is preventing me from going to my maximum photosynthesis rate. Okay, at the beginning, there's not enough light, but as I start to gradually add the light intensity and I move along the x-axis, I notice that my rate of photosynthesis increases, okay? And that's why I'm going to cross out here and here, okay? Another reason why it's definitely not A is because you guys see how the lines of the dotted lines and the straight line is the same, okay? They're on the same line. No matter the CO2 concentration, they are on the same line. So therefore, CO2 is not limiting in this case, okay? Nice. Let's go ahead and look at Y to try to analyze whether it's CO2 concentration or temperature. Now, the key thing is a little tip for you guys for paper 1B. I know you guys love to ask questions for paper 1B. And a tip we always tell our crash course students is to do what? If they give you a a graph question on paper 1B, on paper 1A, on paper 2, okay? And they don't give you information that's not on the slides here, you ignore it completely. Okay, please don't try to make up conclusions on your own head. A little tip that actually that was really useful that my IB biology teacher gave me is that try to act stupid when you're reading a graph question and try to not make any conclusions about any knowledge you know. Because I know that temperature can affect photosynthesis, okay? But if I was like, if I removed all my information about photosynthesis briefly from my head and I just analyzed this graph, I wouldn't have known that. I wouldn't have known temperature could have affected photosynthesis. The only thing that I can clearly see from this graph is that there's a difference of CO2 concentration, and therefore my answer has to be C. Okay? So maybe this was like a really easy example to apply this method, but I promise you, if you try to apply that method when they're asking you to deduce something from a graph, of like trying to not use your outside knowledge and try to just read exactly what's in the slide, it will be really, really helpful for paper 1B. Okay? Now, let's have a look at this one. I'm going to go ahead and scroll up. If I've missed your question now, I'm just going to make sure that you guys have time to read this one and think of the answer. So the table shows information on the genetic code for 4 different mRNA sequences. Which sequence demonstrates degeneracy in the genetic code? Okay, I'm going to go ahead and scroll up while you guys read this. My school submitted almost all my IEAs. Nice. The deadline for IE was Feb last year. Oh, really? Yeah, but there's generally like a school internal deadline and then the actual IB deadline. I just did my language B oral. Oh, that's so nice. Hopefully it went well. My language B oral was kind of deadly actually. I did not like my language B oral. It was so freaking humbling. Mm-mm-mm. And it was so embarrassing because my teacher thought I was going to do much better and then I was like I blanked out. Whew, orals are not for the weak. Yes, internal deadline, but IB deadline's coming. Yeah, exactly. Okay. Has anyone thought of 6? What are we thinking? Okay. Okay. So we have a bit of mixed answers in the chat. How do we answer these questions? Okay, I'm going to go over it. So for those of you saying A, C, and D, try to get into the habit of also, maybe it will be helpful for you guys now since your exams are coming up. Don't only say the answer if you want, of course. If you just want to say the answer, go ahead. Who am I to tell you what to do? But what could be really helpful is you say the answer and you explain why as well. Okay? So if you're seeing that, I'm taking too much time explaining something, try to explain why as well. Okay? We have to memorize. No, for this one, you generally have to see, you have to look at the mRNA sequence. So generally what you have to memorize is you have to memorize the complementary bases. I just know AUG start and met. Good, someone said it perfectly. So, I don't know your name. I want to shout you out, but I don't know your name. Julia? Hopefully I said that right. So you said D because degeneracy means different codons code for the same amino acid. Exactly. Very good. Good job. Where do we see that example? So D is the only time we see that example. And they've been very clear to show us what? They to show us repetition of amino acids here and here in option B. Okay? And so that we can see that the only option that shows different um, mRNA sequences here that code for two of the same is option D. Okay, so because we have GUU here, which code for valine, and GUG, which codes for valine here, we know for a fact it's going to be D. Okay, the reason why it's not this one or this one is because we don't see any repetition of the amino acid, the mRNA base sequence. Oh, okay, perfect, nice. Exactly, good job. Thank you guys for the 13K likes. Okay, someone said, I just know AUG is the start. You do need to know that. Be sure to memorize that as well. A mutation, which type of cell should be inherited? So, inherited means it's passed on from mother and father, so like from parents to offspring. And it says beta cell in the pancreas, T cell in the lymph, sperm cell in the testes, skeletal muscle in the diaphragm. So, from this, you need to know the difference between somatic cells and wait, why is it not B? Good question. So the reason why it's not B is because do you see how the same mRNA sequence codes for the same amino acid? Here it's the different mRNA sequences that code for the same amino acid, and that's the difference. Okay. Nice. We have a C in the chat. We have a D. We have a C. Okay. So a bit of a mixed answers. Nice. We have another C. Okay. So good job. We need to know the difference between somatic and germ cells, germline cells. And essentially, if there's a mutation that can happen in the beta pancreas or something in the T cell or in the skeletal muscle, um, that means it's generally not inherited. Okay, so because it happens in the sperm cell, and remember the sperm will kind of its outer membrane will fuse with the egg cell and release its chromosome inside the cell. The any mutation in the sperm cell in the genes of the sperm cell will affect the child because the child is made up of those chromosomes that they got from mom and dad. Okay, that's why the this answer is correct. Now, for example, me, God forbid, but I could have a mutation in the pancreas now. Okay, a random mutation, or I could even have one in the muscle cell. Okay, but that doesn't mean I was born with it. Hopefully that made sense. Okay, nice. The electron micrograph shows part of a eukaryotic cell. What is the organelle labeled X? Wow, I really hate these. Have you guys seen that meme of like someone making fun of how like IB uses the worst electron micrograph micrograph images? But hopefully this is not too hard. Yes, it's so blurry. Oh, like blurry is my fault or like blurry as in the IB is blurry. Yes, someone said I can never identify the cell structure from the micrograph images. I honestly get you. I think the only way you can improve by that, like for example, in our slide set for the crash course, we have like mitochondria and then we show you different electron micrographs of it. Okay? And I think that will be the most helpful way. No, live is blurry. Oh, so sorry. Do you guys do physics? So we don't have a physics crash course currently. We just added the math crash course. So next year we're definitely going to be adding physics. So if you're M27, hopefully we do physics by the time you um like have to do exams. Okay, we have B's. We have oh, someone said the IB and then someone said the live. So it's both. Oh no. Yeah, I don't know what's blurry. Hopefully it's not blurry anymore. So we have some B's in the chat. B, B, B. Okay. So you are correct if you said B is the answer. The reason why this is the smooth endoplasmic reticulum, guys. First of all, mm, let's first of all cross out some things that are obviously wrong. So, for example, lysosome. Do we know how a lysosome looks like? Lysosome is like our digestive organelle. Of course, I can show the options. What year is this? This is SL time zone 1, November 2025. Exactly. Very good job. A lysosome is like circular. Okay, so then we can cross out C because here I'm sort of seeing some sort of flat discs. Okay, now a nucleus. Do we know how a nucleus will look like? Will it look as well like flat discs like this? Exactly, very good. Ooh, Joya's eating it. You're eating it up. She's being very specific. I really, really like that. Maths, when is it? So the math crash course is going to be after our chemistry crash course. So currently we're doing the biology live crash course. And we're actually continuing tomorrow. So if you want to join that, go ahead and join on the website before you lose the chance to do the live. If you still want to join and you don't want to do the live sessions, don't worry. You have access to the recordings for the next 2 years. Wait, I'm just trying to calculate when the math crash course is. Or let me just go on the website. Okay, so how would a nucleus looks like, guys? A nucleus is also dark, very good, circular, but it has a double membrane. Exactly. So generally, a nucleus of a cell will be large. It will take up lots of space. It will be dark, not necessarily always dark, because remember you have to know the difference between heterochromatin and urochromatin. If you're HL, if you're SL, don't worry about that. Exactly. So that's how we know D is incorrect. Now, the reason why I know it's not the mitochondria is because this is spanning a huge amount of the cell, and the mitochondria has a specific like circular structure, well, oval structure that essentially binds or bounds these cristae. So you might confuse these for cristae, but the reason why I know that they're not cristae is because you guys notice how this like takes part of a large amount of the cell, and it's not like enveloped by a double membrane. Is this past paper available on IB Docs? So IB Docs is down now, but you can just search up IB Docs mirrors. Search up IB Docs mirror, and then you'll have a bunch of links for past papers. For whoever was asking when the math crash course is, it's on April 8th. Okay? Hopefully that is helpful. Okay, nice. Let's have a look at this one. It says which type of protein uses energy from ATP to transfer particles across membranes against the concentration gradient? Hopefully, this is a good and easy question. Hopefully, most of you guys get this right. If not, do not worry. I don't see these past papers. There's only a few from like 2024 and not by time zones. Oh no, which link are you using? Maybe message me. If you could message me on TikTok, I'm more than happy to send you the link. Don't worry about it. But after the live session, okay? So good job if you said B. B is the correct answer. So whenever we're thinking about which protein, so which channel protein allows us to transfer against a concentration gradient, we're thinking of active transport. And the most common example of active transport is the sodium potassium pump. Okay? The sodium potassium pump protein. That's how you know B is the answer. So aquaporin is actually just a channel protein. You don't need to know this for SL, don't worry. Another example of a channel protein. Remember, channel protein is an example of passive transport. It does not require ATP and it goes along the concentration gradient. Glycoprotein, completely different. Who knows what a glycoprotein is? Okay, I think I'll sign up for math. I need to pull a generation lock-in. Yay, that's so exciting. I'll see you there. Can you please help me with like synapses? I'm so confused. Sure. So generally in this live we don't do topics. We don't review topics because it can get really like hectic. But if you're interested in doing topics, I would really recommend you check out the crash courses. We teach the whole syllabus in 5 days, and we go into loads of detail. I don't see anyone from the crash course now, but if anyone here is hidden in the and that has been doing the crash course and wants to give any like review on how it's been going so far, we'd really appreciate it, and give like a full honest review. No one's putting a gun to your head. Oh, I don't know if I can say gun on TikTok live. Cell recognition, exactly. So generally, what you have is you have a transmembrane protein that goes throughout both of your phospholipid bilayers. And then you have a glycoprotein, so a carbohydrate that's attached to your protein. So glyco for carbohydrate that's attached to your protein, and it's exactly used for cell recognition. A protein with a carbohydrate chain extending outside, usually used for. Whoa, Joy, girl, are you an examiner? I think Joy is a secret examiner, guys. Exactly. Good job. If I don't get crash course, can I get personal tutoring with you? You can, of course, but my availabilities are filled to the max, but you can join a waiting list. Okay, just go ahead and join on our website and you can join the waiting list, and of course you can get private tutoring with me. You can get private tutoring with all the other tutors as well. I literally just reviewed this topic today. Yay! It's good. It's perfect that we're reviewing it now. All right. Exactly. We have other tutors on the website. I think, sorry, they were just being specific because they said me, but we have lots of tutors in the website. Okay. Which specialized cell has the largest volume? By the way, guys, if something is not making sense, just let me know and I'll go ahead and explain it again. don't hold out. Like, this is the time to ask as many questions as you can. No question is stupid, because if you don't ask the question now and it shows up on your exam, you're kind of, you know, I was gonna swear, but I don't want to say that. Okay. Someone said, I bet you are full. You're so much better than my teacher. That's so kind. Thank you so much. Which one had the smallest volume? Was it the white blood cell? My teacher is making it so confusing about drawings we need to draw. Oh no, that's so upsetting. In the crash course, we have actually a list of all the things you need to draw for your exam. So basically, the crash course is really helpful because we what's it called? We like create summary sheets and cheat sheets for everything you need to draw, everything you need to like all the long answer questions, all the paper 2 long answer questions. We have a document for that as well. Okay. So egg cell and sperm cell. Which one can we clearly cross out between the cell and sperm cell? Because I think that one's a lot. Um, Maybe you can show the sheet of the drawings. Of course I can. I'm more than happy to show you that. Exactly. So the small. Oh yeah. The smallest one is the sperm. Good job. So the egg cell has to be very large. Does anyone know why it has to be large? This is testing you guys' knowledge. If you're HL, you should know this. Can I tutor for EZ7 after I graduate? Of course you can. We're actually working on expanding our like tutoring section on our website so many people can join and so that you guys can also help other IB students. Yolk for nutrients, perfect. So the egg cell, we must know that it's much larger than the sperm cell. I don't remember how many times larger, but it's actually a big difference. That's because what you'll end up learning is that whenever the sperm basically gives the chromosomes to the egg cell, the sperm doesn't fuse fully with the egg cell. It just, the parts of it here fuse minorly, and then the chromosomes enter the egg cell, and then now you have 46 chromosomes, and then this is degraded. But basically, what I'm trying to say is most of the nutrients come from the egg cell, and that's why it has to be large. Exactly. Exactly. Oh, someone said polar bodies. Yes, exactly. That's why the egg cell is large, because whenever you have meiosis, you have the cytokinesis. You end up having polar bodies, which are just used as waste. Okay, good job. So we know, for example, um, which one has the largest volume? It definitely cannot be B. Now, in comparison of a red blood cell, white blood cell, and an egg cell, the answer would be A. So the egg cell is largest. The red blood cell is small, and I think the red blood cell is smaller than the white blood cell. So it would have been between A and D, and A is the correct answer. Okay. Are these lives posted onto the website? We can post them on the website. We can post them on the Crash Course Hub. I'm not sure if this is the same Uma that's in the Crash Course, but we can, of course. What are polar bodies? A very good question. Can anyone answer in the chat what polar bodies? Yay! Okay, hi. Doing Crash Course right now, going really well so far. For me personally, help with organization a lot. Yay! Don't know if that review helped. It helped so much. I think it's like really it's I am a post crash course student. Yeah, like I was an old crash course student, and now I tutor for EZ7. So it's very hard like coming from the company and being like, guys, please do these crash courses. They help me so much because I work for EZ7. So it's like, is she being dead ass or is she lying to us? So when I see like honest reviews like this, um like I hope those are helpful for you to guys see it. Okay. Does anyone know what polar bodies are? Should I answer? Thank you guys for 20k likes. Wow, I thought we were going to get to 10k, but you guys have made it to 20k. You guys are the best. How is only one egg made during meiosis, even though it makes 4 cells? Good question. That's a really, really good question. So does anyone know what polar bodies are, or should I explain it? So essentially what polar bodies are, remember meiosis, you have 2 divisions. So you start with 1 cell and you split it up into 2 cells like this. Okay, so these are my 2 separate cells. And then you have another division where you split it up into 4. So that's meiosis, and that's what happens in sperm cells. Now, you need to know for your exam, and specifically because they added this and highlighted it in the new syllabus, is that there's something called unequal cytokinesis. So cytokinesis is the process of splitting up the cell after meiosis. Now, unequal cytokinesis is whenever your essentially your cytoplasm and all the organelles are not split equally in between the 2 divisions. So here, for example, in unequal cytokinesis, one of the cells will be very large and the other ones will be very small. And that's what actually you end up seeing when egg cells are created. Whenever you have meiosis of egg cells, whenever you have that second division, one of the egg cells will be picked, and only one of them will actually fully mature to become an oocyte, an egg cell, and the other ones will be called polar bodies. And these polar bodies are used as waste. Okay, they're just like recycled, and their nutrients are reused. Um, and they're broken down. So generally, you have these polar bodies, and they're important, of course, because we need to separate the chromosomes. Okay, but because the egg cell needs to be so large, just like you guys correctly mentioned earlier, um, that it needs to be large for nutrients. Okay, most of the baby's nutrients start from the egg, because remember the sperm doesn't fully fuse. And so the egg cell has to be very large. So you have these polar bodies as a side product. Did that make sense? Would you like me to explain it again? It's recorded. We can also put it on our YouTube channel. Yes, a lot of you guys are like wondering like how can you get like if you want free resources for Easy 7, I would really recommend just tuning in on our lives, following us. Sometimes we post past paper working out on just TikTok, like cute little TikToks of pop quizzes and stuff. And we also, if you want free resources, we have past paper working out on our YouTube. Okay, did that make sense? For the polar bodies, can I move on? Okay. Nice. It says the image shows. Okay, good. Thank you, Dylan. It says the image shows a neuron. Which letter shows a dendrite? Now, this, I think, hopefully, you guys still get this. Okay, you guys are getting it. I'm very, very proud of you guys. The reason why I was a bit iffy is because they're showing a bipolar neuron instead of a unipolar neuron, but you guys are getting it exactly. A is the correct answer. What do we know? Let's do some active recall. What is D? Because you do need to know these structures. What is D? Who knows? Do you need to know how to draw a neuron? You do. Do you guys want me to show you the list of all the things you need to draw? And you also need to label. So draw or label, or you could get a question like this. Okay? Myelin sheath, very good. Exactly, good job. The Schwann cell. Ooh, someone went extra on us. Yeah, it is the Schwann cell that makes up the myelin sheath. Then you have B, which is the axon. Okay? And then you have C. Can anyone tell me what C is? Hopefully C is not too difficult. Thank you guys for the 23K likes. Can you show us the list, please? Of course I can. Let me just take a second to pull it up while you guys are answering. The soma, exactly. It's the cell body of the neuron. Good job. Okay, I'm gonna show the next one while I'm just opening that list of all the things you need to draw. Okay, give me a second, please. This one says red blood cells are transferred from solution 1 to 3 or 4. The image shows the effect on these cells. This is a common question. One similar question showed up on the May 2025, but this is also has just been showing up in previous years as well. Okay. Which year is this past paper? This is November 2025, so the most recent one. What are we thinking for this one? Who knows? And we have access to it. So for these additional resources like the lists and the cheat sheets, you have to be part of the crash course. But if you mean, can you have access to this past paper, this annotated past paper? Yes, of course, I can send it to you. So basically, there's this drive that I've just sent to, I think on the bulletin, I've put on the bulletin where I upload the annotated past papers. So if that's what you're referencing, um, I can send you the annotated past paper, or you can just look up IB Docs Mirror. Thank you, Minna, in the chat. Exactly. Oh, so everyone's fighting in the chat. Someone's saying A, someone's saying D, someone's saying C. And then join the bulletin on our TikTok if you want the drive. Yes, please, can I send it? Of course, just message me, can you DM me? And so I remember to send you the drive link. If anyone wants the drive link of the annotated past papers for free, just message and I'll send you the link after. Um, okay, I'm gonna go ahead and answer this. Sorry, guys, I got sidetracked with the opening the list. So I know that there's definitions of hypotonic and hypertonic. So hypotonic, what does that mean, guys? Hypotonic. And the way I tell the crash course students is what hypo has an O, and therefore, what are we talking about when we're. Oh, okay, someone's remembering something different. When cell explodes. Oh, okay. Okay. So generally, the way I remember it is hypotonic. Because it has an O, I remember low solute. Okay, what does that mean? Low solute solution means that I have a cell. So in this case, my red blood cell. This is my red blood cell. And I have a solute solution. So this red blood cell is in the solute solution. And therefore, I have low sodium outside or low solute outside. So I'm just going to write low Na plus. When I write Na plus, I'm just referencing solute. Okay, perfect. Hypertonic, I remember, is the opposite. So hypertonic means high solute. Okay, so I'm going to go ahead and draw my other one quickly so you guys are like visualized because I'm a very visual learner. And so I always annotate my past papers like this, and I always annotate my exam like this to make sure I don't make any silly mistakes, and I really recommend it. Thank you guys for 25k likes. You guys are amazing. Okay. Oh, the Minna already sent the crash course tutor did put the link in the chat. Thank you so much, Minna. Okay, so now we have the high and the low solute. Okay, we know from osmosis what happens. Water follows what? Does it follow solute or does it follow what does it follow? Does water follow water or does it follow what? What do we know? We know by osmosis, water follows, good job, water follows solute. So essentially, if there's high solute in my solution, that means all the water that's in the cell is going to leave to try to go to the high solute. So this will shrink or shrivel up. Oops, wow. Shrivel. Now I want you to be very, very careful in the terminology we use, okay? So generally, if you say the cell will enlarge or the cell will get bigger or the cell will shrink, be very careful because they've used that terminology in May 2025 and actually it was the wrong answer. So think of swell up, think of shrivel, because actually the cell size itself, like the cell in itself, does not get bigger. The volume does not get bigger in terms of like the cytoplasm. We're just talking about the cell enlarging or the cell shriveling up. And the reason why I'm specific about this is because I've noticed that thing and I don't want you to get the mark wrong, okay? So use the word shrivel and use the word enlarging, okay? Perfect. So swelling, shriveling, enlarging, rather than the cell gets bigger, okay, and the cell shrinks. Nice. So the cell will shrivel when it's in a hypertonic, and then the opposite will happen when it's in a hypotonic. The cell will get bigger. Oh, I just used the wrong terminology. The cell will enlarge if it's in a hypotonic solution. Okay. So solution 1 is hypotonic to solution 4. Now, why is this incorrect? Who knows? Well, because solution 1 is isotonic. Isotonic means what? That the solute concentration has reached an equilibrium. Okay? And so that there's no movement of water in or outside the cell. Now, if this is isotonic and solution 4 we know is hypotonic, and we just established that hypotonic means low, that means I went from equilibrium to an even lower one. Okay? I went from a medium level isotonic to a lower one. Okay? And so therefore solution 1 is not lower to solution 4. That's basically what it's saying. That's why A is incorrect. Did that make sense, guys? Can you show us the list of the drawing, please? I will, of course I will. Just give me a second to solve this so I don't disrupt people who are just here for the questions. Solution 2 is hypotonic to solution 1. So solution 2 here, we have what? We have a hypertonic solution. How do I know? Because I see my cell shriveling up. My cell is shriveling up, and we're saying that it's lower to solution 1. That's not true. We know that solution 2 is higher solute concentration. That's why my cell is shriveling up. That's why B is incorrect. Solution 3 is hypertonic solution 4. So solution 3 has higher solute concentration than solution 4. Let's have a look. Solution 3 here, we have another isotonic. Okay, solution 4 here, remember solution 4, if the cell bursts like this, it means that the water is going inside and therefore it's hypotonic. So we go from a isotonic equilibrium, an average level of solute, okay, to a lower level of solute, which is hypotonic, and therefore it is hypertonic. That solution 3 has a higher amount of solute. Did that make sense, guys, or would you like me to repeat? I know there's lots of hypotonic, hypertonic. So essentially what I want you to do is whenever they're referencing it like this, you can just cross out hypotonic. Okay, made sense, perfect. And just write lower, because we know hypotonic essentially means low. Yay, makes sense. Okay, I'm glad. Okay, solution 4 is hypertonic to solution 2. Why is incorrect? Well, remember this means high, so you can just replace it with high, higher solute concentration. Solution 4 has higher salt concentration than solution 2. No, remember solution 4 is hypotonic. Okay? Nice. I'm going to go ahead and show you this one now, and then I'm going to pull up the list of the drawings. Give me 1 second um, to just orient myself. Hopefully you guys are reading 13 while I'm just pulling up these ones here because I have to download them from the website. Maybe see, okay. Oh, what did I do? I didn't fully understand. You did not fully understand the osmosis, the previous one, or this one. C maybe, okay. Nice, guys. I like that. Even though you're not confident, you guys are saying maybe, you're still answering, which is all that I need of you. B, okay. All right, I'm just gonna, would you guys mind if I just share my screen now and show you the, oh, this is not what I want to show you. Hello? Yeah, okay, nice. Let me show you now, quickly. So here you go on our website here, and then, wait, my screen is showing the wrong thing. Okay, there you go. So you go on the website. Okay, here's your website. Hopefully you guys can see that. And then, oh, okay, there you go. You go on here, you go on your dashboard. Okay, you go on your dashboard, you can go on your crash course. Once you go on your crash course here, you have your crash course hub. You have your day introduction, your day 1, day 2, of course, your resources, all that, all the recordings that you want to watch, whatever, you know. And then where you find the drawing, the list of drawings, you go to additional resources. And then these are the additional resources. So of course we have slides, we have resource packs, we have practice questions, but these are like additional resources that will help you take that step further. So now I'm going to show you exactly what I mean by that. Like, what does she even mean? I mean this. Um, Hopefully you guys can see this now. I've changed it. So this, for example, shows you the list of all the things you need to be able to draw slash label. So if you see this, you might see this labeling in the exam here like so. Okay, and it has the list of all of them. It's like a 30-page document with all the ones that you might see. For example, there's a cycling question actually in the HL exam. And then here are the ones that you need to draw exactly. Okay, we also have lots of past paper analysis. Okay, so what I do, it's my specialties. I just go through a bunch of past papers because I'm just used to doing lots of past papers. Um, and I just like bring up both of them. So basically I've analyzed all the time zones in this one here. Okay, I can also show you the cheat sheet. Those are our additional resources, by the way, guys, besides the slides and besides the resource packs and the practice questions and so on. So these are how can you summarize things that you need to know. Um, like for example, you let's say you've watched the whole crash course and you're like, oh, how can I take this a step further? You look at the cheat sheets to try to summarize everything. So for example, we made this last year for the M25 and it was so helpful. And I think actually most of these graphs showed up in either paper 1 or paper 2. This one showed up as a paper 1 question for HL, for example. And we've compiled all the lists of graphs that you need to know. The meme is low-key relatable. It is. This is the one I was talking about earlier, by the way, if you stayed from the beginning of the live. Yeah, so all these graphs that you need to know for your ecology unit, just the ecology unit, and it's 15 pages of graphs. So that you guys don't have to like waste your time reading a book, making your own notes, going through the syllabus, which is like 100 something pages document. We've already done that for you, okay? So if you go through all that, maybe I can show you a cheat sheet as well. Would you guys be interested in that? Let's see. So, for example, basically how we've created the cheat sheets is that people will tell us like, hey, I don't get this question, this concept in the crash course. Can you make us a cheat sheet? And we make you a cheat sheet. And I don't know if you guys pay attention, but do you remember question 1 of the past paper? It was asking about the water properties. And the water properties, You need to know them with the ring seal. Remember the blubber question and the black-throated loon. Those are the ones that you need to know for your exam specifically. And so we've created this cheat sheet for you guys. Okay, for the person that was asking for the cheat sheets and the drawings, hopefully that was okay. If you want access to these, you just go on our website and you can join our crash courses. You get them additional to the slides. Um, for anyone who's in the crash course and can give like advice, your own advice, how are you finding the slides and how are you finding the resources? Damn, I was only gonna buy math, but Loki might buy bio as well. I actually think bio crash course is the most insane pull. Actually, no, chemistry was actually crazy for me as well. Me doing chemistry was crazy. I got a 4 on my mock exam for chemistry, and then I got a 7 in all my papers, like paper 1, paper 2, paper 3, because I did the crash course. I actually will get the crash course, I think. Yay! Any advice for actually memorizing the content? Yes, Uma. So I'd really recommend that you do flashcards. You buy a whiteboard if you can, and you try to do some active recall. We're in the process of making more flashcards as well and uploading them on the Crash Course Hub for you guys. The slides are great. Yay. I think honestly the Crash Course slides are have every single thing you need to know. Like fuck the cheat sheets, fuck everything, slides, every single thing you need to know. And I stand by that. And I'm so confident because just looking at the slides, um, yeah, just looking at the slides and comparing it to past paper questions, I think we ate with that. Anyway, table shows the binomial naming of 4 species. What can be concluded? Let's get back to this. Yes, active recall, I really, really recommend getting a whiteboard. Like, for example, let's say you have the slides up, Uma, and you look at the slide set, you have a whiteboard up, you hide the slide, and you try to draw what you've just seen on the slide 3 times. Okay? You repeat, you move on to the next slide, try to draw it 3 times, and you cycle through that. Alternatively, I would just recommend flashcards. Flashcards are the best, especially Anki, which is what we have, which is like an active recall special flashcard program. Okay, some of you guys are saying A. Nice. Thank you guys for 32K likes. You guys are the best. I'm so excited for you guys who are thinking of joining the crash course. That's so nice. Hopefully, I'll see you in tomorrow's live session. Okay, most of you guys are saying A. I think some of us have given up, or some of us are still thinking. That's okay. Some of us don't know. That's fine as well. So, remember that binomial naming is actually they removed a lot of the binomial naming that you need to know for from the old syllabus and the new syllabus. So keep that in mind when you're doing past papers. But what you kind of need to know is that as you go from the binomial naming system, and the way I remember it, which is I don't know how you guys like remembering it, but I remember it like Katy Perry came over for grape soda. Okay, there's also an inappropriate version, but whatever you like to use. As you go from kingdom to species, you get more and more specific, okay? And as you go up to kingdom, you cover a broader range of organisms. And so the key idea is that here we notice that the evolutionary origin is similar or closely related between some of them because they have a similar species name, or as well, you can think of it as a genus. So A is the correct answer. Good job. Well, actually, A is not the correct answer because the genus would have to be the same. So actually, let's have a look. Yes, W and Z are the same ones. You have Felice and Felice, which is the genus name. Okay, therefore C is the correct answer. So careful, guys. Remember, kingdom is the main one, and it's the overarching. Okay, and you go from kingdom to species. So kingdom, phyla, you have to remember all of them. And so basically, the higher up you are, so remember there's genus before species, the more common the names are going to start to get. Okay, and if you have an organism or species here that has a same genus, we know that they're more closely related. So for your exam, you might be like, she's talking too much. What do I need to know? What you need to know is genus. If you see genus and it's the same, it's closer in relation. That's what you need to know and you need to write down and memorize. Okay? Nice. All right. But aren't species closer, like more specific? Actually, no. Good question. No, it's about genus. So you need to know that genus, they're similar, they're closer related, and what we mean is historically or evolutionary wise. Okay? So Hopefully that made sense. I think genus means genetic, so they are genetically similar. Ooh, okay. I like that. I like that memorization technique. Did that make sense, guys, or can we move on? C, yes, yay. Yes, okay, nice. Let's move on. Thank you guys for 33K likes. Ooh, I hate these questions. This is part of the new syllabus. Oh no, I was scrolling up too high. This is part of the new syllabus. New syllabus. So it's good that they have given us now practice questions that we can provide you guys. What are we thinking for this one? This was also showed up on the paper two, where they ask you to name like the epidermis and the parts of the leaf that you need to know. So here it says the image shows the transverse section of a leaf, which letter identifies the palisade mesophyll. Yep. Okay. Perfect. You guys are saying B. Anyone have any objections? How are you guys feeling, by the way? Are you guys feeling tired? Do you guys want to end the live here, or are you guys wanting to keep moving forward? Do you guys want to see more of the resources? Do you guys have any questions about studying, about IAs, about EEs? Dylan said keep going. Anyone have any other objections? What do you guys want to do? I'm honestly okay to keep going for a bit more. Studying questions. Now is the time to ask questions, guys. Yeah, move forward. Okay, nice. No, let's keep going. Yeah, just a bit, it's mainly 2 a.m. now. Oh no, keep going. Okay. I'm gonna upload. I'm actually right after this. I'm gonna hold myself accountable, and I'm gonna figure out what's wrong with our YouTube. I'm gonna upload this live exactly on our YouTube, so that you can watch this. By the way, if you need to go to bed. Yeah, thanks so much. I'm serious. No video has helped me. Even my teacher says thank you so much. I'm really glad that we could help. I really appreciate it. You guys are more than capable. Like we have the same experience of having horrible. Bro, when I used to ask my chemistry teacher for a question, he would laugh out loud in front of the class. It was bad. So I get you with the bad teachers. That's the goal at Easy 7, to just help you as much as we can, provide free resources when we can. This is the free resources we can provide you, to try to provide you authentic resources that are actually good quality and that, can help you get your final grade. Someone said my teacher, Minna said my teacher made us watch PewDiePie during bio class. Bro, why are teachers so bad? I have no idea, especially for this new syllabus. I have no idea. Ciao. I have no idea. I spent 20 minutes every class doing a meditation. Okay, you guys are correct if you said B. Good job, guys. That is the correct answer. That is the palisade mesophyll. You just need to know that. You just need to label it. Um, and if you want, we're gonna cover this exact topic tomorrow. So join the crash course while you still can. This is topic 6 of our crash course. I think my teacher isn't IB certified. I'm literally live in a place with 4K people. Oh no, yeah, I don't think most teachers are actually IB certified. Oh, my teacher told me I shouldn't study chem if I'm bad at math. Girl, I'm genuinely shocked. This is the motivation that teachers are giving you. That's a lie. I think you can more than study chemistry. Even if look, yeah, I kind of suck at math, even though I'm the math tutor. So I get you. Chem math isn't even that advanced. Yeah, I agree. Okay, this is what is the property of veins. This is a common question. It has been showing up previously. Yeah, I would not stress at all. Don't worry. I love when you say girl. I didn't even notice I was saying that. A, okay, I like that you guys are speedy with this. A, nice guys. So we know veins are unelastic. We know arteries are elastic so that they can um, change the volume and withstand high pressures. Okay, because what is the point of arteries, guys? Do arteries carry blood away from the heart or towards the heart? What does fenestration mean? Good question. So maybe do you speak what languages do you speak? So I can help you out. For whoever asked, sorry, I don't know your name. Your name is just user 799 and 444. Um, what languages do you speak? Do you speak maybe French? Any Latin? Away, good job, guys. Arteries move away from the blood away from the heart. So if you can think that my heart is up here, okay, on my left side, and I need to move blood all the way to the tip of my toes, okay, it must have such high pressure in order for me to push the blood all the way to my toes. Okay, and that's why I think that arteries, because they move away from the blood, there's high pressure, and they need to have um, like elasticity so that they don't break. Okay, that's why B is incorrect. We know that's not for veins. Now, I was trying to ask you, sorry, if you just speak English, that's more than fine. I was just trying to give you a way to help you memorize. So fenestrations, maybe you know the word in like French fenêtre, or like, yeah, fenêtre just means window. Um, and so you can think of it as like your blood vessel has a bunch of little windows. Okay? And that's what I was trying to get you to do because it will help you memorize in the future. Okay? Nice. So, fenestrations for rapid exchange. Um, mm, that's not generally a property of veins. That is a property of capillaries. Okay? And so that's more specific to capillaries. Narrow lumen to carry deoxygenated blood. It does carry deoxygenated blood. Does it have a narrow lumen though? Who knows? You need to know like a list of the characteristics about the lumen, about the elasticity, about the muscle layers, about the thickness of the walls. Very good. It has a wide lumen. Generally, and you also, by the way, need to identify in a micrograph. Okay. And so generally what you'll see is that a vein will look kind of floppy like this versus an artery, which will have a very thick wall and a very narrow lumen. So it actually has a wide lumen. That's why my answer is A. Okay. Nice. They're usually in the artery near the kidney, I think. So actually, no, careful. Fenestrations are not in arteries. They're in capillaries. Okay. Isn't flexible and elastic the same thing? No, not in this context, but very good question. So flexibility just means I can kind of like bend it and like squeeze it and like squish it down and really like conform the shape, deform the shape of the walls. Elasticity means that I can for example, like think of muscle that you can extend and then contract. So that's what we mean when we say elastic. Elastic means that you can reach a specific point, but then re-reach your origin again. And so that's what elasticity means in physiology. Okay? So elastic just means going to a point and then reaching that same point back. Or maybe if you do physics, that will be helpful. Like a rubber band. Thank you, Ali. Yeah, exactly, like a rubber band. So that's what elastic means. Flexibility just means I'm deforming the vein in this case. Okay, nice. Um, exactly. Someone also mentioned the wall. You do need to know the wall, so good job that you mentioned that. Which is the receptor in a pain reflex arc? So actually, guys, this is old syllabus, kind of, because they didn't use receptors and like effectors and um effectors. There's one. Wait, effectors and what? No, no, no. I have to remember this. Effector. So effector is the. I'm not gonna give you the answer. There's no way I'm forgetting this. Good job. So most of you guys are saying, oh, actually, yeah, good job. So most of you guys are saying D. Nice. So what? Who knows what an effector is? Yeah, that you can use effector and sensory. Nice. Effector is the organ that reacts to receptor. Exactly. Good job, Joyce. Nice. So remember that there's a receptor and an effector. Sorry, I'm trying to remember that other term I'm remembering off the top of my head for receptor or sensory. Exactly. So something senses the pain stimulus, and then something your muscle moves your hand away from that stimulus. So effector is a muscle, and actually there's a question in the HL paper, I think in May 2025, if I remember correctly, and the other time zones of November. So keep this in mind. They've repeated this a lot in the time zones. And that's the effector, so that's the muscle. So we can already cross out this because we know that's not the receptor. So what is receiving that pain stimulus is going to be a sensory nerve ending. Okay, gray matter and white matter is a new concept as well that they've added to the syllabus, so I'd really focus on that in case they show it up. Pituitary gland, we know that's just an endocrine organ that releases hormones. Okay, so that's why that's wrong. Which function is primarily associated with helper T cells in the immune system? I hate the immunity unit with a passion. Even when I'm learning it in uni right now, I also still hate it. How do you guys feel about the immunity unit? Despise it, yeah. Me too, honestly. We have C's and B's. Don't like it either. Yeah, I really hate it. I don't, especially like adaptive and innate immunity. One thing we did do though is we predicted, guys, we predicted the M25 paper two that they were gonna ask a adaptive immunity question with that special graph that you need to know. So be very careful. Familiarize yourself with the list of graphs that you need to know for immunity, so that you don't lose any marks. I did normal bio last year and loved it. Oh, okay. I think it just depends. Immune system is tough. I agree. Okay, so some of you are saying B, C, B. So you are correct if you said B. Good job. I'm just gonna go ahead and quickly grab some water, guys, because I've been speaking for a while. It's just off the screen right here, okay? I'll be right back. Give me 1 second. All right, I've got my water. Perfect. Okay. So, what is the function primarily associated with helper T cells in the immune system? So, phagocytosis of pathogens. Does anyone know the actual white blood cell that is responsible for this? Like, which immune cell is responsible for this? Good job if you said B. Does anyone know off the top of their head? It's actually not T helper cells. It's not lymphocytes. Have you guys heard of macrophages? Maybe. Good job. Good job, Ali. Macrophages. Exactly. Macrophages. They engulf. Okay, that's their whole purpose. Good job, guys. Macrophages. It engulfs. It phagocytoses. Cytose. Hopefully, I use that correct. Um. And so basically, it engulfs or like chomps on pathogens. And so it's not A. Antibody production. No. What produces antibodies? Who knows? Retention of memory cells. There's a other cell that also looks like this. Good job. So B cells are responsible for memory retention. It's not T cells. T cells are specifically for the activation of other cells in the immune system. Good job, guys. So the B cells hold the memory, and then they go to T cells, and then they tell, hey, T cell, activate the immune system, and then T cell activates it. Okay? Nice. This, I think, is, yeah, this is a common question that has shown up in time zone 1, time zone 2. I think those two time zones of M25. And now it showed up on November. So what advice am I going to tell you? Make sure to cover this very well. What what are zoonoses? Who knows? We've just covered this in the crash course, I believe. They are diseases. Actually, no, we have not. Have we? I can't remember. Um, but they're essentially diseases that can be transferred directly from animals to humans. And I gave you the answer, but most of you guys are getting it correctly. Yes, the answer is A. Okay, they these are examples. You actually do need to know a couple of examples or recognize a couple of examples, and we have them on the crash course slides. Um, and essentially, yeah, they're just diseases that can be transferred from animals to humans, not from human to human. Zoos, this is just trying to confuse you. Antigen to animals, no, it's just animals to humans. Okay. Okay, let's have a look at 19. A cross section of a flower is shown. What is structure? Why? Diseases cross from one species to another. Kind of, because animals are different species. Like you can think of an animal that's a different species than a human, even though humans are animals. But I feel like you get what I'm trying to say. Wait, I'm missing on something crazy in the chat. My teacher gives stupidly hard tests. There was a question about obligate and facultative parasites. Is part of the syllabus? Yes, it is. It is part of the new syllabus. Careful. It is. It's new to May 2025. Okay, it says a cross section of a flower is shown. What is structure? Why? So you need to know actually stigma and an anther, and you need to know how fertilization of um a flower is shown. You also need to know about self compatibility. I'm just trying to list the things that can help you guys study. It's on the SL syllabus. What's on the SL syllabus? The obligate anaerobes. I think yeah, obligate anaerobe just means yeah, it's on the syllabus, on the SL syllabus, just means that it can't survive on oxygen conditions. You do need to know that. So most of you guys are saying D. That D is not the correct answer, is it? Ah, it is the correct answer. Nice, guys. So you need to know the anther and the stigma. So this is the stigma. And if you look at the list of all the things you need to memorize for the syllabus, this is one of them. That not list of all the things you need to memorize, the list of all the drawings you need to do. This is one of them that you need to be able to like recognize and label. So make sure to have a look at that. Um, um, yeah. And so generally it goes from the anther to the stigma, and that's how essentially fertilization occurs, where you have pollen here, and the pollen goes here, and then you have fertilization here. Okay, perfect. Let's have a look at question 20. Ooh, I like this question. I always like inheritance questions because I used to suck at them. I used to be so bad, actually, like dead ass. I used to be really bad at these questions. And then I joined the crash course, and Minna, who has been responding in the chat, actually taught me it. And now I love these because I always get these right. So hopefully you guys get this right as well if you've done the crash course. If not, that's okay. We're gonna explain it together. Oh hell no. No. No, guys, please. Keep up the energy. I never get these. No, 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 no. You're gonna get it today. Guys, Eleanor is my icon. She put C in the chat. How was the crash course inheritance topic? If anyone has done the crash course inheritance topic and is getting this question correct, I'll take that 0. Um, let everyone know how the inheritance topic, because I feel like I actually am the one that taught the inheritance topic. And I feel like most of the students in the crash course understood that, which is really good sign, or kind of got a good idea for them to know how to approach their future study. Okay, let me know what you guys think. So one person just answered that, and if this is the same as my student, then I think she's in the crash course. Yes, especially the question pack I grind on. Yay! Okay, I think it's the same student that's in the crash course. Sometimes I like mix up the names and I see someone, oh, you're in the crash course, and they're like, no, I'm not. I'm like, oh, sorry, I recognize a different name. Yes, I am. Okay, yeah, yay, and you're the only one that got it. You're the only one that got it correct. And then also, PSJ, which is previously mentioned, is in the crash course. C, yes, good job, guys. C is the correct answer. Yay! I'm happy. I'm happy. I'm proud of you. But don't worry, guys. If you're in the chat, join our crash course, or I'm gonna explain it now. Okay, or do both, whatever. But the pedigree chart shows the incidence of color blindness in 3 generations of a family. Color blindness is caused by a sex-linked recessive allele. Okay, so if you're doing the crash course, are we doing ecology tomorrow? Yes, we are. Someone said best ad. I'm really not trying to make this an ad. Like this is supposed to be a free um session. So, but yeah, I get what you're trying to say. Anyway, okay, so you need to know the sex-linked recessive. You need to know the sex-linked dominant. You need to know the autosomal dominant, and you need to know the autosomal recessive. Okay, and you need to be able to identify them in an allele, in inheritance diagram. You need to be able to make conclusions on them. Okay, so essentially, if something is a sex-linked recessive disease, what does that mean? It means that you must have which type, which phenotype. Can anyone tell me? Let's say, for example, the letters we want to use is R and small r, big R and small r. Those are the letters we're gonna choose to do. I haven't started studying yet, and it's stressed, but I got like 80% of these questions right. Am I good? Ooh, um, hmm. I think it would depend on your like pattern. Like, are you getting these correct by luck, or are you also? Did you notice that you're throughout the year you've been getting good, um, like good grades in biology? I think it would depend on that. Like maybe it's just, maybe this is a good past paper. Maybe you know this stuff. Even if you haven't started studying yet, I think you're still good. I just would recommend that you start now or start soon at least. So I didn't mean that. I meant like an ad as in like life proof. Yay. Okay. 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 Good. 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 Good job, guys. R all good. Don't worry. All small r. Very good. I have been getting 6 and 7 on tests. Okay, that's a good sign. Wait, I'll reference you back again. Let me just work this question now with the students. Exactly. So good job, guys. It's gonna be small r, small r. So basically, essentially, you need to have both small r's in order to have the disease. Okay, to be affected. So we know that this male is definitely affected like this. Uh oh, I've used small r when I should be using X and B. Fine, sure. Because it is sex linked and it's on the X chromosome, whatever. So the patient who's affected should have large X recessive, large X, which is this basically just represents the chromosomes, and they have the recessive alleles. Okay, nice. Let's go ahead and write this down. Nice. And we need to find out what his sister has. So if it's recessive and someone's not affected, but I see that the parents have passed it on to the child, that must be that mean that the parents are what? Who knows? There's a specific word that you have to know, specifically with recessive diseases. That very good job. Exactly. Carrier. Exactly. Good job, guys. Carriers. So it means that the parents must be carriers. In dominant diseases, you don't have carriers, okay? It means you either have affected or non affected. But good job, guys. So these parents must be carriers. So they must have that recessive allele like this. I see that here, but they're not affected. Okay? And since I've concluded that, this question becomes really easy. Because what can I do? I can just draw a Punnett square. Who remembers from the crash course? Who remembers from the top of their head? Which goes here? Is it the male or female that goes at the top of the Punnett square? And well, who goes on the side? The male or female? So on the top, it's the mom or pop, mom or the female. Good job, exactly. Here it doesn't matter because they both have the same. They both have the same um, alleles, so it doesn't really matter. But in case they ask you to do a Punnett square in your exam, you make sure to get the mark fully. Um, okay, nice. So then you have XB X large B, X big B, small b, X large B, X small b, and then you have X BB. Okay, nice. So the reason why the answer is C is because our individual set Z could be X large B, or this individual Z could be X large B, small b. Now, can anyone guess why this one we didn't take it into account? Why did we not take into account this X small b small b? What do we see? Do you guys notice something? Since he's not affected, good job. Since this is not colored in black, we know he's not affected, so there's no way he has the homozygous recessive. Okay, that's why the answer is C. Did anyone understand? For everyone who was getting or was like not answering or was maybe said the wrong answer, that's okay. No worries at all. But do you guys understand why it's C now? Do you guys feel a bit better about this? Because the circle is in black. Yes, exactly. Female unaffected. Exactly. Yes, makes sense. Perfect, guys. I'm really, really glad. I think this is actually quite an easy inheritance question. I think they could be much, much ruder, and they have been much ruder. Actually, there was a May 2025 one that was really rude, if I remember correctly. Yeah. Okay, I'm just gonna scroll up to see if I missed anything. Thank you guys for 47K likes. You guys are the best. Let's see. It says the results were obtained by measuring the body temperature of people exercising on a hot and humid day. Okay, guys, we're on question 21. We're 9 away from the last question. I think we can just push through to try to finish this whole past paper. And then I will upload this on YouTube. Oh, I remember I said I was gonna go up and respond to someone while I was mid answering. Who was that? Mm. Oh yeah, so you were saying how you're getting sixes and seven on the test, and you got 80% of these questions right. Am I good? I think you are good, but I think that you should start studying. Maybe if bio is not your priority, you can just start doing a little bit every day. You can. I wouldn't just say you're good and like forget about it because, hmm, like if you have, like I don't want to tell you don't study, you're fine, and then before the exam you're like, oh, I should have started studying. Anyway, it doesn't harm to start studying now. Wait, could you please repeat question 12 after? Of course I can. My teacher uses in thinking for the test, and the questions are awfully hard, and these past paper aren't. All right. Yeah, especially for math. If you guys need any helpful tips for math, I would really recommend. Oh, you'll upload this live on YouTube. I will. I will upload this on YouTube. For math, a lot of students are like, I don't know how to start studying for math. I don't know how to do this. And actually, lots of people, guys, I'm so sorry. Lots of people on TikTok, like old IB students who are like posting on TikTok stuff. Oh no, people are leaving as soon as I say I'm uploading this on YouTube. Yeah, I will be uploading this on YouTube. Thanks for sticking along, guys. Whoever's still left in the chat. Um, but a lot of people, a lot of old IB students, like who are getting 45s or who are getting whatever, are saying, if someone's telling you to do past papers, don't do them. Just do this and this and this, and then they're like selling something. You all do not listen to that advice. That is the worst advice someone could ever give you on IB. I don't care what they're selling you. They could be selling you, I don't know what they're selling you. Do not listen to that advice. Do past papers. And the point I was making is that even if you just study 20 minutes every day of a past paper for math, and then you slowly build that up to like 20 minutes, 40 minutes, an hour every day of math, you're going to see so much results. But the important part is you must start now, and you must start like little and gradually build yourself up for it. And that's really, really important so you don't get burnt out and so that you don't regret not starting earlier. Okay. So do a little bit every day. Like, I'm being so for real. If you could just do like 3 questions of a past paper, and then the next day you do the other three, and so on, it will help you so much until you can fully lock in after your IAs are done and you have much more free time. Okay. C, because vasodilation and temperature are too high, but we need to go to the surface, lose heat. Yes. I'm literally skipping topical questions and skipping to past papers, hoping it covers the syllabus. Yes, I think. Oh, it depends what you mean by topical questions. Like if you're saying from the question bank, then I think they're really helpful. But past papers are the key thing. Yeah, exactly. You have a mix of time, so you should do a mix of both. But closer to your exam, you should focus on past papers. Okay? What I meant by like don't listen to people who are selling you other things, I mean like yeah, people who are selling you, I don't know. I don't know where I was going with there kind of, but just promise to do past papers, guys. These results were obtained by measuring the body temperature of people exercising on a hot and humid day. What mechanism could result in the changes seen during rest period? Okay. So here, exercise, your body temperature is increasing, and then rest period, your body temperature is decreasing. Increasing, decreasing, increasing, decreases. Decreasing. Now, during exercise, your body temperatures increases. You can kind of use your past knowledge with this, like briefly, okay? Because it's not asking us to conclude anything from the graph. Remember when it's asking you to conclude something from the graph or tell us what can be deduced from the graph when they use that terminology directly, or they ask you to analyze what relationship you see in the graph, you have to kind of take your previous knowledge out of this. But in this case, you can kind of use previous knowledge. So after you exercise, do you shiver? No. Okay, so A is incorrect. Uncoupled respiration in brown adipose tissue. What is this process? Who knows? What's the function of brown adipose tissue and the uncoupling respiration? It's to cause a what? An increase or decrease in blood. Very good job. It's to generate heat. Okay, and so what we're seeing here is that you're resting and your body temperature is decreasing. And actually, this B cannot be the answer because the whole point is you're trying to analyze how does your body decrease its temperature back again? How do we go through these cycles? And it's definitely not by generating more heat with brown adipose tissue. Okay, C is the correct answer. If you said that, good job. Vasodilation of the skin. Someone worded it perfectly. Okay, someone said, I'm going to scroll up now. I think it was Joya. She said, C, vasodilation. When temperatures get too high, blood needs to go to the surface and lose heat. Exactly. So when your body temperature gets too high, your blood vessels start to vasodilate. And so essentially they start going from like this size to this size. And so that you increase your surface area so that more heat can reach towards the surface. Imagine this is your skin, and so that more heat can leave your body. Okay, and that's how we allow for this rest period where your body temperature decreases. Um, Now hair erection just means that you're like get like goosebumps, or like when you see like physically see your hair lifting up. Okay, and that generally happens when you're cold. Okay, nice, perfect. Let's have a look at this one. And this one actually was just in the this was in our topic 1 slides. I just saw this, by the way. This exact example, this exact freaking example. Um, and it says bonobos and chimpanzees are primates. Separation by the Congo River allowed these two species to be formed. What is a consequence by the separation? This was also a paper 2 question, by the way, in May 2025. So lock in, guys. Remember, separation. Nice, you guys are getting it correct. Exactly. This is an example that you need to know for your exam. Most of you guys are saying B. Good job. It is reproductive isolation. So, what is a definition of a species? Who knows? Who knows? There's a specific thing that you have to know for your exam to define species. They have to produce what type of offspring? Exactly. Fertile. Good job, Dylan. Good job, guys. Good job. Organisms that can produce fertile offspring, fertile offspring. Perfect, guys. You got it. And so this example that you need to know is that they essentially had reproductive isolation. They were isolated. Why? Because the land that was separated by the Congo River prevented them from mating together, and then that caused new species and evolution. And that's what they're saying. Okay. And now, because they're a new species, what happens? They cannot produce fertile offspring, and therefore, this allows us to have um, two new species being formed. Do you guys notice something? Do you guys notice how the genus is the same in reference to what we talked about in one of the earlier questions, but the species is different? Okay. And what did we say that the genus being the same means? Who knows? The genus being the same means that they are similar, right? They are similar and they're more likely alike. So keep that in mind. We see that exactly more similar. We see it being brought up again. Good job, guys. We're almost done. It says what could minimize the loss of biodiversity? So biodiversity, minimize the loss. So what could we do to prevent? I hate when they like are saying minimize the loss or decrease the loss because I'm like, wait, what? Decrease, increase, loss. Yeah. I always start tweaking out, but it's actually not too bad. Minimize loss. So how do we prevent less biodiversity? Nice. So you guys are saying D in the chat. Does anyone have any objection? Whoa, we hit 51K likes. Thank you guys so much. I really, really appreciate it. You guys are the best. D, B, okay, D. Nice. We have different answers in the chat. I like seeing a mix. So D is the answer. Um who knows what rewilding is? Rewilding is part of your new syllabus, and we're going to cover it now in topic 6 of the crash course. So if you still haven't joined the crash course, by how many times I've freaking mentioned it in this live, but we're going to be covering this whole ecology unit. I know a lot of you guys got the ecology unit self study, which is actually not cute at all, and you had to self study in the summer. So this is the perfect time for you guys to join because we're about to teach what rewilding is. Who knows what rewilding is? So deforestation, deforest, you're breaking down forests, you're breaking them down, cutting down the forest. If you increase the rate you are breaking down forests, you're losing more biodiversity. So this is just doesn't make no sense. Introduce species into new environments to control predation. Actually, no. Introducing species into new environments would actually can cause more loss of biodiversity, right? That's why they have so many laws of like what plants you can bring on a plane and so on and so on. Nice. Someone said building up ecosystems that have been destroyed by humans. Perfect. Exactly. So that's a really good definition of rewilding, and that's how we can minimize the loss of biodiversity by trying to rebuild those ecosystems that have been destroyed. Okay. Reduce the use of renewable energy. Again, why on earth would that minimize loss of biodiversity? Why is IB playing with us? I think this one's obvious. You try to increase the use of renewable energy. That's what you would see in your exam. Good job, guys. What is evidence for evolution? Common question, common old syllabus question. We got this in the bag. Dogs and wolves has been a example that you need to know. Inheritance of acquired characteristics, domestication of dogs from wolves, homologous structures such as pentadactyl limbs. What are homologous structures? Who knows? Homologous versus analogous structures. We just did this in topic 5. You guys are almost done. Thank you guys so much for the 52K likes. You guys are so amazing. Keep up the good work. Don't lose motivation, guys. It's been a long live. We've been live for, I think, almost 2 hours now. No, not almost 2 hours. We've been live for 1 hour and a half. There you go. I just checked. Nice. Same origin, but different structures. Perfect. And analogous means different origin, but same structure. No, I'm losing it. Analogous means, for example, a wing of a butterfly versus an insect versus a bird. Okay. So you have different origins, but it has the same function. Now, homologous structures, you have the same origin but different function. There you go. Exactly. Thank you. Analogous have same function but different origin. Nice. And so there's an example of the homologous structure that you need to know. The most famous one is the pentadactyl limb. Okay. And is that evidence for evolution, guys? Do we know? Yes, right? That isn't an evidence of evolution that you need to know. Exactly. So I is correct. So if I is correct, that means I'm going to cross out C. Domestication from of dogs from wolves. Is that evidence of evolution? Yes, good job. Exactly. Nice. So I'm going to cross out B because it doesn't show option 3. So now at this point, you have 50-50% chance of guessing it. Yes, it does. Good job. This is an example you need to know. Domestication of dogs and wolves and artificial selection. You need to know that as well. Inheritance of acquired characteristics. No. Exactly. Very good. That's Lamarckism. That's exactly the other theory that is proposed. This one's incorrect, and that's why the answer is A. So you don't have the inheritance of the acquired characteristics. Do you guys remember? Maybe you learned it in class because you do need to know this for your syllabus. Basically, there's this other theory that believed that, um, like how giraffes were created or how giraffes were evolved is that they started like eating all the leaves at the bottom of the tree, and then they couldn't reach the top tree, so then they started having longer and longer necks. Okay, and that's what this is referencing. Okay, and this is incorrect. It's not the inheritance of um, acquired characteristics that is evidence for evolution. Perfect. What describes holozoic nutrition? I also hate these. I hate the chemoautotrophic, photoautotrophic, holozoic, sapotrophic, tetravore. They're not too bad, but I just hate them. Nice. Someone said D in the chat. Anyone have any other ideas? Guys, we're almost there. Keep it up. We have 5 questions left. We're at 53K likes. You guys are amazing. I can't wait to see all the new people that join tomorrow. And I can't wait to see the familiar faces of all the people who were there from last time. What are we thinking? D, D, D. I think D is the correct answer. Good job. D is the correct answer. So, halozoic nutrition, it's key because we need to know that essentially there's difference between external and internal digestion. What does that mean? So, essentially, saprotrophs, they have these enzymes which they release outside of their body, and these enzymes digest food and they break it up, and then they absorb the remnants of that food. Okay, and so that's why this is incorrect because it's actually supposed to be um other way. And then heterotrophic has internal digestion. That is true. How does that happen? It's like just like us. For example, we have digestive enzymes in our body, in our mouth, in our stomach. Okay, and then we absorb it in our small intestine. So we have internal digestion, and then you can have external digestion. All right. Hopefully that made sense. Which interspecific relationship has a negative impact on both individuals? So interspecific. So the way I like to remember is inter has an e, and so it's between. Therefore, between what? Between species, right? And it has a negative impact on both individuals. Then we know it must be what? Okay, some people are saying A, good job, exactly. Competition is when they're both competing, they're both fighting for the resource, whether that's land, whether that's food, shelter, whatever, they're both have a negative impact. Parasitism, one benefits and the other one doesn't. What is mutualism? Who knows what mutualism is? Do some active recall in the chat. Even though it's late, you guys got it. It's kind of in it. It was like a mutual agreement. Both benefit. Exactly. Good job. Whenever you have a pathogen, um, you have like it's kind of like parasite actually, the pathogen one, when you have like a host, and then one benefits and one doesn't. Okay, nice, good job, guys. These both benefit. I just plug in my computer to charge. I'm getting a bit tired, but we only have 3 questions left. We got this. The image shows a food web from Chesapeake Bay. Which organisms are tertiary consumers in at least one food chain? Okay, you guys have this. I feel like I brought my water and never drank it. So I'm also going to take a water break in a second. What are we thinking for this one? When they are both using the resource to help each other. Nice, mutuals. Perfect. It's a good example of mutual. Good. Oh, it's kind of blurry. Thanks for letting me know, Dylan. Is this better? Zoom in, please. Of course. Is this better or more zoom in? I think you guys don't need the question, right? Like I can just go like this. Okay, I'm gonna leave it like that. Perfect. Okay, good. Thank you guys for 53K likes. Oh, my mic. What are we thinking? Kind of saw that in the chat. I also hate these questions. Okay, we have a B. So tertiary consumers, and in at least one. Generally don't remember, don't worry about it. Don't worry about it at all. Primary consumers are what? What are primary consumers? Think of the food chain. Who knows off the top of their head? Anyone? So you have primary consumers, and then you have secondary consumers, which eat the primary consumers, and then you have tertiary consumers, which eat the secondary. Exactly, very good. Organisms that eat producers, exactly. That are feed on autotrophs, exactly. That feed on photosynthetic. So we know that plants are photosynthetic. They're autotrophs. What does that mean? That means they make their own food. How do they make their own food? Via photosynthesis, and all these things you have to know for the syllabus. Gosh, I hate IB. And then we know that the um, primary producers are the plants. Then you have secondary consumers, which eat the primary producers. And then you have a cycle, and then you end up having the tertiary consumer, which eats the secondary consumer, and da 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 da. Okay, now let's have a look. So, vegetation means they're referencing plants. Okay, aquatic vegetation, just plants and water. Got it. Phytoplankton. Got it. Phytoplankton also undergo what? Photosynthesis, therefore they're also autotrophic. Got it, okay? And then I'm kind of seeing the primary consumer, the secondary consumer, and the tertiary consumer. Okay, nice. So, which ones are tertiary? We see the small planktivorous fish and the Tundra swans. Let's first identify those. Oh, wow. I didn't realize how much analyzing I was going to have to do. Okay, I found the Tundra swan. She's here. Where is the other one? Ah, there is small planktivorous fish. Okay, we notice that they go from straight from the vegetation, straight from the plant to the specific highlighted ones. So there's no way it's tertiary because tertiary would be like the third in the in the food chain. Okay, herbivores, ducks, and bald eagles. Let's have a look. I'm gonna highlight in a different color so you guys are able to recognize this easier when you're looking at the annotated slides. Remember, if you want the annotated slides, I can send them to you after. Send you the drive link, or you can scroll up in the chat to find it. So herbivorous ducks are here, and bald eagles are here. Okay, so this is looking like the correct answer. Why? Because I'm just looking at how many arrows there are. Okay, there's 1 arrow, 2 arrow, 3 arrow here to represent that this is a tertiary consumer. So 1, 2, 3, and then here it's 1, 2, 3 as well, and therefore actually B is the correct answer. Wading birds here, we see that it's secondary, so it's not this one. The geese, where's the geese? The geese is primary. Okay, the osprey, where's the osprey? Here it's tertiary, yes. Let's have a look at this one, but this one's primary. Okay, this is why this is incorrect. Perfect. Let's have a look at the next one. This is the diagram represents the carbon cycle, which arrows represent respiration. Nice, this is not too bad. Can you repeat? Of course I can. So essentially what you have to look at is the tertiary consumer is going to be the highest in the food chain. And so you have to see how many arrows, or how you basically have to count 3 arrows that take you from the primary producer to the tertiary consumer. Okay? Does that make sense, Joyce? Or do you want me to re-explain again? Let me know. Okay, let's have a look. The diagram represents the carbon cycle. Which arrows represent respiration? What do we know? So from plants to animals, from plants to fossil fuels, from fossil fuels to CO2 in the atmosphere and dissolved in water. What also causes CO2 in the atmosphere and dissolved water? Who knows? Respiration has products of what? Cell respiration has products of CO2. Okay, nice. Someone said why for sure. Yes, exactly. And so the answer is quite easy. Why? Because we know that B is the only one that has Y. Okay, but good job for identifying that it's Y. How do we know? Well, we know respiration is the process of combusting glucose, which is taking glucose and burning it in excess oxygen to, or also not excess oxygen, aerobic or anaerobic, to make CO2 plus water. Okay, and so actually a product or a source of the CO2 in the atmosphere and water is from animals that undergo respiration. That's why Y is correct. Let's have a look at T. Well, we know this arrow here, because you also need to see this arrow. This arrow has to be photosynthesis. Why? Because we know that taking up CO2 and water from the atmosphere and taking it into green plants is the process of photosynthesis, where you're actually reversing this exact equation and making up glucose. But you must remember that as well, plants undergo respiration. Okay, that's why T is also respiration. Nice, exactly. CO2 and water are the reactants of respiration and the products of respiration and the reactants of photosynthesis. Okay, a bird of paradise. This is also slay, guys. This is also in the topic 5 crash course slides that we just did. Literally, that's what we ended on last weekend. Okay, don't ever play with our slides. We always give every single one. The reason why I'm so excited is because I this is the first time I'm seeing this past paper, and so I'm liking that these examples were on the slides for M25 and the November 25 crash course. Okay, a bird of paradise has very showy plumage and demonstrates a unique dance. What type of selection pressure could have caused this plumage to evolve? Nice, you guys are getting it. Yay, most of you guys are getting it. Be very proud of yourself. I just did this question this morning. Nice. Okay, thank you guys for 55K likes. I really, really appreciate it. Isn't V photosynthesis too? Sorry guys, I just saw your yeah, exactly. V is photosynthesis. Okay. D is the correct answer. So you know how you have to know that basically that they do this special dance and they have these like very very large and blue striped feathers. Like this, they're all black and they have like a section of blue striped feathers because they are trying to attract specific mates. Okay, so this is uh, the reason why this selection pressure evolved is to attract specific mate. Okay, and that's how they're basically these movements have forced them to it's added pressure on them. It's forced them to attract specific mates. I don't. I've described that really, really weirdly. I'm so sorry, guys. I'm starting to get tired. Um. Yeah. Do you want me to re-explain that? Let me know. Or does everyone get this in the chat? Yeah. I love how in animals it's always the males trying to attract. It's actually very cute to think about. Maybe animals are romantic. Makes sense. Okay. Nice. Okay. Let's move on to the last question. Oh no. Why do we leave the most deadly question to the end? That's okay. We'll do it. So that shows how the hardiness zones in part of North America are predicted to change over the next 25 years. A hardiness zone is an area that has a certain average annual minimum temperature, a factor relevant to the survival of many plants. The lower the number, the more cold resistant the plants must be. I have no idea what I just read. Hopefully you guys are locked in. What is the likely consequence of this change of tree species? Okay, let's process this because I think I did zero processing. So it's saying the data shows the hardiness zones in parts of North America and how they're predicted to change over the next 25 years. Fine. So this is the hardiness zones, then how they're predicted to change over the next 25 years. Okay, a hardiness zone is the area that has a certain average annual minimum temperature. Okay, certain average annual minimum temperature. A factor relevant for plants. Why is it relevant? Because when you have a low number, the more cold resistant the plants are. So they survive better in cold environments. Okay, now I kind of get what's going on. This is what is a likely consequence of this change for tree species. So why do over time, so back in 1960 and 1990, what do we see? We see, remember, the lower the number, the more cold resistant they are. Okay, so the better they are at the cold. And what do we see? We see that it's generally only here. And then what do we notice? We notice a pattern that the highest number here, okay? But then you also see here from in the next 25 years, the highest number, which is 11, increases. Okay, so the hardness zone 11 increases because I'm assuming you have less resistant plants to the cold. Why is that the case? Good job, guys. You guys are getting what a complicated question for such a simple answer. Exactly. A is the correct answer. Why? Oh, oh no. These are my slides from uni. Sorry, guys. Um, yeah, exactly. Because we have climate change, which is causing temperature changes to the earth, and therefore there's less cold resistant plants. So you see the lower the numbers are here, the low numbers here are kind of disappearing, and more of the I'm sure the opposite of cold resistance, hot resistant plant is starting to increase. Okay. Wait, what topic is this on the syllabus? This is topic 6. Don't worry, we're gonna do it tomorrow. With climate change and um, yeah, so what you'll notice is that the last few questions here are like evolution and topic 6. Okay, by the way, when I say topic 6, I mean the crash course is topic 6. For you guys, it's just ecology, but for our crash course, we call it topic 6. From which country are you streaming from? I'm streaming from Italy. Which country are you from? Just to know what time zone this is for. This, Italian time zone. Oh, you're also from Italy. That's so cute. Oh, wow. Look at us, twins. Okay. Oh, Palestine. What other countries? Toronto, Spain. Oh, wow. Look at us. We're international. Sweden. The one good thing about IB, it's international. Qatar. Wow. France. That's so cute, guys. I don't know why I'm getting so sleepy all of a sudden. Does anyone have any questions after today's session? Thank you guys for 60K likes. You guys are the best, honestly. I hope the session was helpful. It was kind of informative one. Thank you. Of course, it was kind of a good one to do, I think. Especially like I don't know. It was kind of good to see because I think from like a tutor's perspective, it was nice having like ideas confirmed from May 2025 because you can analyze May 2025 and then when you see the same pattern in November, it's kind of confirming and gives me more confidence in teaching you guys and creating more resources. You're very, very welcome. I'm really glad this is helping you guys. I hope my teacher uses this mock. I hope for you as well. When will you go live again? So I have the crash courses on the weekend, so maybe Monday or Tuesday. I'll try to go live around then. I'll try to text on the Easy 7 bulletin. Remember, guys, if you want, will you solve tomorrow? I unfortunately not because we have the crash courses and we'll be teaching in the crash course. So if you're interested in and you like this type of teaching and you're like happy with what Easy 7 is doing, follow us, follow the bulletin board, or join our crash courses, whatever you guys want. What is the YouTube channel? Easy 7. I think it's just called Easy 7. No, I forgot. Oh God, my one job, and I forgot it. Give me a second. I think it's called just Easy 7. Um, or just search up Easy 7 IB Sciences. Yeah. Thank you so much. Super, super helpful. You're very, very welcome. I'm really glad I could help. Um, yeah. Easy 7. It's called Easy 7. Yeah, it's just called E7. It's called E7 Sciences or E7. And I'll upload it right now. Does anyone have any questions? Does anyone want to see the Crash Course Hub? Does anyone have any questions about how to study? Thank you guys for 61K likes. You guys are the best. Questions? Thank you guys for staying up with me. I think we haven't done a full past paper in long. Your Anki deck is so good. Yay! I'm glad. I think actually Minna made the Anki deck. So you can thank her for her iconic work when she did the Anki deck. Thank her tomorrow. Yay. Okay, cute. Okay, any questions? Yeah. another cute addition is that you get Anki decks, which are like flashcard decks when you join the crash course. Thank you for all the gifts, guys. I really appreciate you. No questions? Okay, so now what I'm gonna do now is I'm going to upload this annotated slide on the drive so you guys can have access to this annotated slide. And I'm gonna check out your DMs to see if any of you guys need the link. Then I'm gonna go ahead and straight upload this to YouTube so you guys can use this. And then I'm gonna work on some crash course resources, and actually I'm gonna work on some past paper analysis for November 2025 so that the crash course students can benefit off this, okay? I think they might cancel Middle East exams, so I don't know if I should lock in. I will. You definitely should lock in, because imagine you don't and then you regret it. Just lock in, and whatever happens in the future, I'm sure like you won't regret studying. Like I don't think someone could ever regret studying, because maybe they use your mock. Imagine they use your mock predicted grades. Hopefully they don't. Actually, I would be so stressed about that. But better safe than sorry. Exactly. All right. Thank you guys so much once again for 61K likes. I really, guys. I hope. Yeah, I know they're coming up. Maybe your teacher's gonna use your predicted then, or your IA, like they did in COVID. I wonder. Anyways, hopefully everything will go well. Yeah, once again, if you guys like these resources, check out our crash course, check out our YouTube channel for these lives, check out our TikTok for just funny content and more past paper questions. Stay updated, join our bulletin board, message us if you want any resource, and we'll send it to you. And I hope you guys found this live helpful. I wish you guys a good night or a good morning wherever you are in the world, and best of luck. All right, message me if you need anything, just words of motivation, you need help, advice. You can just message E7 and we'll reply. All right, bye guys. Ciao.